Thanks, Boris. I want to thank Boris and Karen Anderson for asking me to come over and talk to you. As Boris said, I'm at NASA Ames, just on the other side of the creek. And I want to point out, first of all, that this talk is a review talk based on many people's work over many years. I want to start with talking about why is the search for life interesting. And I'll give two reasons. One is that if we find evidence of what I call a second genesis of life here in our solar system, that will allow us for the first time to be able to do comparative biochemistry. As you know, all life on Earth has the same biochemistry, the same genetics. We only have one example of life. If we found another example, we'd be able to compare and contrast our life with another type of life. Also, philosophically, it would tell us that life is common in the universe. If right here in our own little solar system, life started twice independently, then we'd be pretty sure that life is widespread in the universe. And this is interesting, and I've added my own little editorial comment. Yay, that'd be a great thing to know scientifically. I guess some people think they know it in terms of alien visitations, but in terms of scientific evidence, we have no evidence that there's life beyond the Earth, and finding it in our solar system would be the first step. In a sense, let me elaborate on that a little bit. In a sense, it's comparing apples and oranges. This is a diagram that Scott Sanford put together for the Journal of Irreproducible Results, comparing apples and oranges. And I like to think of searching for another example of life is doing a study about fruit and only having oranges. It'd be very nice to have apples. If you had apples and oranges, you could do a more elaborate study. And of course, you'd want to have bananas and pineapples and even tomatoes as well. Tomatoes, biologically, are obviously fruits, even though the Supreme Court declared them vegetables. And you want to compare them all. So in a sense, what we are in a position on Earth of having only one example of life, only oranges. We want something different to compare to so that we're able to understand life at a broader level. Where are we going to get a second genesis of life? There's three research programs aimed toward a second genesis. First is make it in the laboratory, what is now called synthetic biology. The second is find it on Mars or Europa. That's the program I'm interested in. And finally, there's SETI, as you heard from Seth. Listen for them to call. It's not clear which of these will work, which one will succeed first. But my own interests are not in waiting for the phone to ring or playing around the laboratory, but going out in the field and seeing if we can find it. And when we say go find it, we're really restricted to our own solar system. And that means we're looking at the other worlds in our solar system and which ones are possible places where life could have started and trying to find evidence for that. And that's what is going to be the focus of this talk. And I'm going to very quickly go through the best worlds in our solar system that might hold example for life, talk a little bit about what we're doing and how we're approaching it. Mars is number one always. Mars is number one for three reasons. Here's an image of Mars as it might have looked in the past, today, and in our imagination, Percival Lowell's map of Mars. Mars is the number one target in search for life on Mars for three reasons. Evidence of past liquid water, presence of an atmosphere with the elements needed for life, and importantly, the potential for preservation of evidence of life. If there was life on Mars billions of years ago, there's a good chance that there's a record of it there. In contrast, consider Venus. Venus might have had a liquid water past. It certainly has the elements needed for life. But if there was life on Venus, there's very little chance that we're going to find a record preserved. Would you rather keep your sample in a hot sulfuric acid bath or in a cold vacuum conditions? Well, obviously, cold vacuum conditions. Mars is where we might find preserved evidence for life. Very quickly, what do we know about Mars? Most of what we learned, what's what we know we learned from Viking. Viking found water on Mars in the terms of frost and found flows that indicated past water. Water now only is ice and vapor, clouds, snow, no liquid anywhere on the planet now, but evidence that there was liquid in the past. That, in a nutshell, is the fundamental story of Mars that we've learned from spacecraft exploration. And, as I mentioned earlier, the elements needed for life in the atmosphere. Here's a CO2-dominated atmosphere on Mars, the same sort of atmosphere we see on Venus, the same sort of atmosphere we think characterized the early Earth. This is the original atmosphere of the Earth. The oxygen that's in our atmosphere now is a fairly recent pollutant. It's only been around a couple billion years. Of course, humans in their wisdom are getting rid of that pollution and bringing back the CO2, restoring the Earth to its environmentally correct initial condition. And many species on Earth, in fact, will thank us for doing that. There's nothing wrong with this atmosphere. It's a perfectly good atmosphere for life. The problem on Mars for life is not the elemental composition, it's the pressure. Here's the pressure on Mars over 3 Mars years. You see it varies from 7 to 10 millibars. On Earth, the pressure at sea level in these same units would be 1,000. So the pressure on Mars is 100 times thinner than the pressure on Earth. And that has important implications for the stability of liquid water. The triple point pressure of water is 6 millibars. The triple point pressure is the pressure below which the liquid state doesn't exist. The reason dry ice on Earth never forms a liquid, which is why it's called dry ice, I guess, is because the pressure on Earth is below the triple point pressure of carbon dioxide. Well, on Mars, the pressure in most places is below the triple point pressure of water, and here at the Viking sites, which are in lowland sites, the pressure is close to the triple point. As a result of that, liquid water doesn't exist on Mars. Water goes directly from vapor to gas, to solid, solid to vapor with no liquid phase. Water on Mars behaves like dry ice does on Earth. Now, that's bad news for biology because all organisms on Earth, the one ecological requirement they all have for life is liquid water. We know of no organism on Earth that can grow or reproduce without essentially being a bag of liquid water. So that's the story of Mars very quickly, bringing you up to date. What are we learning right now from the missions that are going on around Mars? There's 4 missions operating, 1 recently lost. They're giving back more data. You can go on the web and see it streaming in. They're basically reconfirming that story that we got from Viking. More and better evidence for water. This is, right now, I would say the best picture we have from orbit, arguing for a liquid and arguing that that liquid is water. A canyon, the Didi Canyon, a couple kilometers across, a channel that looks like the river that carved the canyon, snaking back and forth. All evidence of flow, repeated flow over a long period of time, evidence of water, ruling out volcanic, wind, ice, and other alternative models for what carved the liquids. Where's the water now? It's frozen in the polar regions, as we thought. Direct evidence from the Mars Odyssey mission shown here. This is the maps of hydrogen and, by inference, water in the ground, frozen in the ground in the 2 polar regions. So Mars essentially has a frozen ocean in the polar regions of both poles. That ice is very interesting. We'll come back to it later. The 2 rovers landed, also found evidence for water in the layered rocks, in the salt distribution, and in these concretions, so-called blueberries. So all pointing toward the notion that Mars had more water in the past. And if we put that all together and take the geological estimates for how much water Mars must have had, we can paint a picture like this. This is obviously an artist's conception of what Mars might have been like 3 billion years ago, when it had a layer of water 500 meters thick, which is what the geologists estimate is implied by the fluvial features. Well, this looks familiar. This looks like Earth. This is the Mars that's interesting. This is why this picture shows why we send more spacecraft to Mars than all the other planets put together. In fact, we crash more spacecraft on Mars than we send all the other planets put together. It's because of this image, because that Mars once had a wet history. It was once a water world. It was once like Earth. We once had at least 2 blue marbles in our solar system. We want to know, or I want to know, was there life when Mars had water? What kind of life? Was it a separate life from life we have here on Earth? And what happened to them? Are they still around? What are they doing? Well, let me start by, why does Mars not look like that today? If we once had 2 blue marbles, why did we lose one? And the answer is not that Mars is further from the sun. That is the incorrect explanation for why Mars is a frozen desert world today. The answer is that Mars is too small. Here are the planets shown to scale, Earth and Mars. Mars is 1/10 the mass of the Earth. A factor of 10 in mass matters, and it changes the properties of the planet significantly. As a result of its small size, Mars has no plate tectonics, less gravity, and no magnetic field. All 3 of these factors tend to cause Mars to lose its atmosphere. As a result of losing its atmosphere, it freezes and everybody dies. If Earth lost its atmosphere, it would also freeze. The mean annual temperature on the Earth, global temperature on the Earth, is plus 15 centigrade. If Earth had no atmosphere, no greenhouse effect at all, the average temperature on the Earth would be minus 15 centigrade. Now, you guys know the difference between a plus or a minus. It makes a big difference. Earth needs its greenhouse effect. Now, we humans are augmenting the greenhouse effect, which is probably not necessarily a good thing. But the fact the planet needs a greenhouse effect, Mars had a greenhouse effect and lost it. because of these factors, all to trace back to its size. I listed those factors in the order of importance that I attribute to them, although there's still some debate in the planetary science community. I think plate tectonics, the absence of plate tectonics, is what makes Mars, what primarily caused Mars to lose its atmosphere. Venus also lacks plate tectonics. This is the cycle of the long-term carbon cycle driven by plate tectonics on the Earth that we think is what keeps the Earth habitable. Mars fails in this long-term cycle because it has no plate tectonics to recycle sediments and carbonate. Venus fails because it has no liquid water to pull the carbon dioxide out of the atmosphere. This concept of plate tectonics maintaining habitability is what you might call the master idea in planetary science. And when we look at other planets around other stars, we look at their size because we think a planet must be at least half the size of the Earth to support this kind of cycle. So an Earth-like planet then, by definition, is a planet that's half the size of the Earth, not more than 10 times the size of the Earth, and is at the right position of its star to have temperatures between 0 and 40 degrees centigrade. And just FYI, as a little footnote, the astronomers discovered the first Earth-like planet that fits in that box outside our solar system. There's now, if we imagine a diagram of mass and temperature and a box for Earth-like conditions, Earth is in that box, sort of by definition. We now have another point in that box, first time. I think that's very important. Back to Mars. Mars is not in the habitable box. Mars is too cold. It's too cold because it's too small. It lost its atmosphere. And we see on Mars evidence that it's been cold for most of its history. This is images of unweathered rocks on Earth, and these are images of Mars. Mars looks like a fresh volcanic flow that's not been weathered. These results from the TESS instrument on Mars Global Surveyor. It looks like a planet with its geological history dominated by cold, dry climate, in which mechanical weathering was the dominant form of erosion, reading directly from that quote. Unlike the Earth, where chemical weathering promoted by water is the dominant form of erosion. So even when Mars was wet, even when we look back at that picture of Mars 3.5 billion years ago with lots of water, it was still very cold. And I just list some of the evidence for that: low erosion, the sporadic distribution of the valleys, the unweathered surface materials, and so on. Mars was a very cold place. And so if we're going to places on Earth that are analogs for Mars, we want to go to cold places and investigate how life survives there. And this image showing one of the rover landing sites, this red X, is a graphic illustration of this phenomenon, what I call the paradox of early Mars, that it was very cold and very wet at the same time. Here is Gusev Crater, a large crater lake. We think this was full of water, 160 kilometers across. We see what looks like a large river that flowed into it, a couple kilometers large. So this is large, the size of the Sacramento River. You could even imagine whales stuck here swimming the wrong way, being chased by a flotilla of boats and helicopters. Large river, large lake, all dried up now, but evidence of considerable flow of liquid. We can even see what looks like a delta deposit where the rivers enter the lake. And yet, in the same frame, you see ancient crater terrain that looks like the moon. You see a moonscape of craters, which is completely inconsistent in terms of our terrestrial experience with large rivers and lakes. Those craters indicate a terrain that has never seen rain. So we have a paradox of early Mars, which is rivers and lakes without rain. Even when Mars was wet, it was cold. And the best place on Earth to illustrate that, the only place on Earth where we see rivers and lakes without rain, is the dry valleys of Antarctica. Here's a Landsat view looking on the coast of Antarctica. This is about 2,000 miles south of New Zealand. Here's the coast of Antarctica. Here is Ross Island, Mount Erebus, the active volcano. And here is McMurdo Station, the main US base in Antarctica. If you look closely, you can see the icebreaker track carrying essential supplies, candy bars and powdered milk, into the station. They won't bring whole milk. This region here, the dry valleys, is the largest ice-free region in Antarctica. It's the most Mars-like place on Earth. Its average temperatures are minus 20 centigrade. It never rains in these valleys. It snows, but it never rains. The biggest difference between these valleys and Mars is that in these valleys, the pressure is well above the triple point of water. But the temperature is not that much warmer than Mars, and the absence of rain is characteristic of Mars. We think this is a model for early Mars when the pressure might have been higher, but it was still a very cold place. This is a place where we see rivers and lakes without rain. This is the mighty Onyx River flowing here. It flows a couple weeks each year during the Antarctic summer. On a cold summer, it might not flow at all. And during a very wet summer, it might be so wide you can't even jump across it. It is the largest, longest river in Antarctica. It flows into this lake, Lake Vanda, one of the largest lakes in Antarctica, 5 kilometers across, 170 meters deep, completely ice covered. The ice cover is about twice as thick as this room. But that's what you expect when they have annual temperatures, -20. So we have large lakes, rivers, in an environment where it never rains. If you were a little organism sitting on the side of this lake here, you would see snow falling down in the summer, and then it would sublime or be blown away. You would never see the liquid phase. Just a few meters away in the stream bed, you would. So along the sides of these valleys is essentially lifeless. Lifeless with very low erosion rates because of the absence of liquid water. Water is only produced during the summer when these glaciers melt, during a few weeks each year when average air temperatures get above freezing, and flows down to power these lakes. So we go down there every year because this is the most Mars-like place on Earth and study the ecology of this system, a system which has very little ecology at all. This is a picture of our camp. This picture is taken on Halloween, which is why we're flying the Jolly Roger, of course. We were trying to invite trick-or-treaters to come to our camp. We had a bowl of candy and everything. We didn't get any trick-or-treaters that Halloween. We think they were waiting for it to get dark. So we set up camp here, and we go out on the lake, and we melt a hole through the ice cover, a couple meters across. As we drill down through the ice cover, it's dry, it's frozen ice, beautiful views looking up inside the ice cover. We get through the ice to the water comes up into the ice. This ice is floating, so the water level comes up to 90% the thickness of the ice, as expected. We then have an access hole for getting into this water. And so we get some volunteers, lower them in the water. Fortunately, NASA's a federal lab, and we have a lot of non-essential federal employees, and so we lower one down. We can't afford the rope. We do have rope, so we tie them to the rope. So this is me getting ready to do a dive a couple years ago, going down, and this is the view going down, and hopefully also the view coming up. Follow the rope back up. You literally can't just free ascent up through a 4-meter thick ice cover. You've got to go back out the same hole you came in. In that sense, it's like cave diving. Well, why do we do it? We're not swimming in this cold water just because it's fun, although swimming in freezing water does have its charms. The reason we're exploring this lake is because on the bottom of this ice cover, or underneath this ice cover on the bottom of this lake, we find a thick mat of organisms growing. Remember, this valley is essentially lifeless. This valley walls contain no life, essentially. It's all cold and dry. But underneath this thick ice cover in this cold water, we find abundant life. Just microbial life, just what, to use a technical term, green slime. There's no fish, there's no tadpoles, there's no anything, just microorganisms.
It is green, so they get some sunlight?
That's right. The question is it looks green, and it is green. These are photosynthesizing. The ice cover freezes so slowly that its optical properties are remarkably pure. And through that 4 meters thick of ice, the transmissivity is about 1%. So the light level in this lake is about the light in this room right now. We do not need flashlights when we swim in this lake, and the organisms can easily grow at that light level, just like that plant over there, which hopefully is not plastic, is growing at the light levels in this room. Algae can't photosynthesize at these low light levels. So this is a photic system underneath 4 meters of ice. Who would have guessed, flying over these lakes? That's the system that we think could have been alive in this lake on Mars. So imagine now, 3 billion years ago on Mars, very cold, an environment where it's not raining, an environment like the dry valleys of Antarctica, which is why these craters are preserved, because rain is not eroding them. But like Antarctica, there is seasonal melting producing copious flow which fills a river and fills a lake in an ice-covered lake. And Antarctica is showing us that cold, wet environments are perfectly good for life. They may not be good for human, warm-blooded life, but for green slime, that's ideal. So we have a good scenario to go and drill in lakes like this and try to find the evidence of that green slime that might have lived in this lake on early Mars when it was a cold and wet place. And Antarctica provides the case study for the logic for doing that. What we might find in a lake like that on Mars is a fossil. But remember, I said we were searching for a second genesis, and fossils are not enough. What a fossil tells us is merely that there was life on Mars, right? But it doesn't really tell us the nature of that life or its relationship, if any, to life on Earth. And that's the question that I'm really interested in. In another way of saying it, is Martian life on our tree of life? Here is our tree of life, as first elucidated by Carl Woese. Based on ribosomal RNA, we can show that all life on Earth is genetically related. We all share a common ancestor, shown here, and divide into three major domains: the eukarya, the bacteria, and the archaea. This is life on Earth. This is the wiring diagram, the family tree, whatever you want to call it, of life on Earth. There's a fundamental unity to all life. We see an apparent diversity. We see blue whales and trees and tadpoles and bacteria, and it looks like there's a wonderful diversity of life, but that diversity is all superficial. It's all morphological. The bricks from which those organisms are built are all the same amino acids, the same nucleotides in the DNA and RNA. There's one blueprint for life on Earth shown here. What we're asking is Martian life also on this tree? Are they related to us? Is Martian life just another branch on our same tree? Do we share a common origin, or is it something completely different, not mappable on our tree at all? It could be that Martian life did share a common origin with life on Earth. For a long time, we thought that life on another planet, by definition, had to be alien life. But now we realize that Earth and Mars are not necessarily biologically isolated. There's been exchange of material between Earth and Mars. We have on Earth some almost 30 rocks that we know came from Mars. Here's a picture of one of them, the famous ALH 84001. That rock came from Mars. How do we know they came from Mars? Well, they have little plaques on the bottom that say, "Made on Mars." Not really. What they have is inside the rocks, they have little bubbles of gas, and the composition of the gas in those bubbles is the same as the composition of the atmosphere as measured by Viking. And that's different from the atmosphere of Venus or Earth or the Moon, which has none, or Pluto or Goofy or any other world you wish to consider, which tells us that these rocks came from Mars. There's other lines of evidence as well. It is accepted that these rocks came from Mars. We also know that these rocks got here by being kicked off Mars, flying through space and landing on Earth. And that this process of ejection and then landing did not heat the interior of the rock hot enough to sterilize it. And we know that from magnetic domains within inside the rock, which have not been reset, and they would be reset at temperatures of about 40 to 50 centigrade. So if there was a little bug inside this rock on Mars, and it got ejected into space and landed on the Earth, it could still be alive. Question?
How long ago was that rock?
Pardon?
How long ago did that rock hit the Earth?
This rock landed on the Earth tens of thousands of years ago in Antarctica. ALH means Allen Hills. It was ejected off Mars a couple million years ago. It formed on Mars 4 billion years ago. So the history of this rock is it's a lava flow on Mars 4 billion years ago. It sits on Mars. About 2 million years ago, an impact occurred which knocked it into space. It spent a lot of time orbiting the sun, and then by chance landed on Earth in Antarctica. And then in 1986, some explorers were down there and found the meteorite and brought it back. So back to imagine that scene several billion years ago when Earth and Mars are exchanging rocks. We know that this process goes from Mars to Earth. We assume that it could go from Earth to Mars. Imagine a rock being kicked off Mars, an organism inside it, landing on the Earth and breaking open. And a bug crawls out and says, one small step for a Martian bug, one giant leap for Martian bug kind. And Earth is colonized by Martian bugs. So we now realize that Earth and Mars may have exchanged biological material. To use the technical term, they may have been swapping spit early in their youth when the impact rate was much higher. And that's bad news for astrobiology, because it means that if we go to Mars and find life, that life may be just related to us. We can't take for granted that that life represents a separate origin. Life could have started on Mars and been carried to Earth, or it could have started on Earth and been carried to Mars. And so the burden of proof is on us if we find evidence of life to determine that it really is a separate genesis. So I want to now turn to that. Fossils are not enough. I'm calling this a forensic investigation. We need a corpse. We don't want a fossil, we want a corpse. We want a dead body. Anybody who watches TV knows that dead bodies are a lot more exciting than fossils. That's what we want to find on Mars. Well, where might we find a dead body? Well, I list the possible sources. And the one that I'm going to talk about today is very subtly put in boldface, underlined, and has an arrow pointing at it, which is that I think our best chance of finding biological remains of Martian organisms is preserved in the permafrost on Mars. Permafrost is a good place to find organisms. On Earth, you all know about Siberia. This is a cross-section of Siberian permafrost. Here's the surface, and then a meter down is the active zone, and below that is frozen ground. In this particular image, this ground has been frozen for 3.5 million years. We find things, frozen organisms in the permafrost a lot. Question?
Can you give an idea of the scale of the picture?
This is a pipe from a drill that the Soviets put in back in Soviet times, and is about 5 inches in diameter. So this is about a meter, this distance here, and the entire cliff is about 10 meters high. And this is a cutaway. There's a river flowing in front of the image here, cutting away at the cliff, which is why there's such high rate of erosion here. In this permafrost, we find organisms frozen for 3.5 million years and still alive, bacteria. We find things that have been frozen for shorter periods of time and are dead, like mammoths and horses. But bacteria, frozen for 3.5 million years, still alive. In Antarctica, we find what may be even older permafrost. Here is ice underneath our camp here, and underneath a meter of till, there is ice that some geologists think is 8 million years old. If so, this would be the oldest ice on Earth. And its temperature is -25. It also contains viable bacteria that have been frozen for 8 million years, if this ice really is 8 million years old. So we're trying to develop the technologies to drill in these permafrost conditions aseptically so that we get a sample that's useful for biology, and we want to then fly that technology to Mars. And we use tracers. You can't sterilize a drill rig, but we can trace its contamination with fluorescent beads that track the contamination from the Earth, from the core. And here is an example where the core interior is clean, the beads have contaminated a very thin region on the surface, and here is the drill with the beads in place. Now let's go to Mars. We want to find the oldest frozen organisms on Mars. So this is a map of Mars showing several things. It shows the crater distribution on Mars. Each crater is marked by a green circle, and you can see that the southern region of Mars, in the southern plains, is heavily cratered. This is presumably then the old part of Mars. The north region of Mars is smooth and presumably has been resurfaced. We don't know what resurfaced it, but presumably the north then is younger than the south. So if we want old, we'll go south. We want ice-rich. These blue lines show the distribution of ice as determined by the Odyssey probe, and the deep lines show the distribution of deep ice as inferred from crater morphology. So if we want old and we want ice, we go south of this blue line. But this map also shows one more thing. It shows the crustal magnetic features on Mars. This is a surprise discovery. Mars has strong magnetized rocks on its surface, and they're shown in polarity, blue for minus and red plus, and you see they cluster around here. There's some here and here. What's interesting about the distribution of this magnetic material is that there's none around Hellas, the giant impact, or around Argyre. It appears that these impacts erased the magnetic material around them. Whatever created the northern plains erased the magnetic signature there, which tells us that two things. It tells us, one, that places that still maintain the magnetic signature have been relatively undisturbed, and two, that they're very, very old because they predate Hellas, they predate Argyre, and they predate the north-south dichotomy. So places that still have the magnetic signature are very ancient and relatively undisturbed. So if we want to find ancient, cold, ice-rich ground that's been relatively undisturbed, it's this yellow X. That's our target zone. Now it turns out we've already landed in the yellow X. We crashed in the yellow X when Mars Polar Lander was sent to Mars in 1998. At the time Mars Polar Lander was targeted, we didn't know about the magnetic features. We did not know about the distribution of ice. It's somewhat serendipitous that it was targeted right where I would like to go to find the ancient ice. But this is where we'd like to go, drill deeply and search for frozen organisms that may be 3 billion years old. Will they still be alive? Remember, on Earth, even in the oldest ice we find on Earth, the organisms are still alive. But that's only 8 million years. What determines how long you can sleep in ice and still be alive? This is a good science fair project. You can do this at home. The answer is probably billions of years. There's two factors that limit survival in ice. One is just thermal decay, what I call KT. This is not a big deal on Mars because it's so cold. The second one is radiation. Just from natural crustal uranium, thorium, potassium at low levels, deep below the surface. It's not cosmic rays or solar radiation. This is natural radioactivity from the crust. In Siberia, it's 0.2 rads a year, which is not a lot. Organisms that are alive can easily repair that level of damage, but an organism that's frozen and inert, it's going to accumulate a lethal dose. Even the champion radiation survivor, Deinococcus radiodurans, would accumulate a lethal dose in 100 million years. 100 million years, well, that's much longer than the age of ice on Earth, but it's much shorter than the age of ice on Mars. The conclusion, the profound conclusion is, it's dead, Jim. Well, of course it's dead, Jim, but dead is actually okay. We're not interested in a live organism, we're interested in a dead organism. And in fact, we'd have to kill it to do the analysis anyway. We want to look at its genetic and biological materials, right? The important distinction here is not between live and dead, it's between a dead organism and a fossil. What we'll find in this permafrost, we hope, is a dead organism. This search will start in a few months when Phoenix launches to Mars and starts landing in the polar regions, digging down to the ice and a few centimeters into it. That hopefully will be followed by a deep drill going down maybe 10 meters, and then an even deeper drill going down 100 meters into this ancient old ice. So that's our best chance for finding evidence of life on Mars. I want to now quickly talk about the number 2 candidate for searching for evidence of life, which is Europa. This is Jupiter, and here's its 4 Galilean moons, Io, Europa, Ganymede, and Callisto. Europa, number 2, the brightest one. You can look through a small telescope, and if you stare at the Galilean moons long enough, you can pick out Europa because it's obviously brighter than the other 3. It's an ice-covered world. We think it's got a layer of water underneath the ice, maybe 100 kilometers deep, underneath maybe 10 kilometers of ice. What's the evidence for the water? There's 2 lines of evidence for the water. One is morphological. We see what look like icebergs on the surface of Europa. Well, icebergs float in water, so this is morphological evidence that this terrain was associated with liquid features. But this could have been an ancient terrain that's frozen solid. If you have icebergs and then you freeze the ocean, it's still going to be icebergs. This is not evidence for an ocean today. This is evidence for an ocean at some time. The evidence for the ocean today actually comes from a very interesting application of what I call airport technology, which is the metal detector technology. You know, as you walk through the metal detector, there's a time-varying magnetic field going on. The metal detector creates a time-varying magnetic field which induces currents in any metal or any conductor, and those currents create a magnetic field which then disturbs the imposed magnetic field and generates a disturbance in the imposed magnetic field, which is detected by the electronics, which is why a metal detector detects not just magnets, but it detects any conductors at all. Well, that same effect is going on with Europa moving around Jupiter. Europa is in orbit around Jupiter. Jupiter has the strongest magnetic field of any planet in the solar system. Europa is moving through this varying magnetic field. As it moves, it's creating a disturbance in that magnetic field, which indicates that Europa has a large conductor on it. Now, presumably Europa doesn't have a bag of dangerous nail clippers with which it's going to hijack the Jovian airplane. What could the conductor be on Europa? The only answer is a global salty water, ocean. That's the evidence that Europa has an ocean today. It's the effect on the magnetic field of Jupiter as it moves through it, the same effect that you induce when you walk through an airport detector. So next time you fly, think about Europa as you go through the security line and the alarms go off. Boris?
What excludes a liquid core of metal?
The question is, could this be explained by a liquid core of metal? The answer is no, because we know the composition of Europa is dominated by ice and silicate from its density, and that even if you separate out the core, it'd just be a little tiny thing. What we need to explain this signature is a large global conductor. And it doesn't have to be high conductivity. It could just be a slightly salty ocean. It can't be pure fresh water, but it doesn't need to be as salty as Earth's ocean, for example. So that's the evidence Europa has water. Given water, does Europa have life? Well, it has to satisfy 2 requirements. One is that it has to have a plausible origin of life, and the second is it has to have a plausible ecology. Well, we don't know how life started. We do not have a consensus theory for the origin of life. We have a bunch of theories, and on timescales of roughly 10 years, the theory in fashion changes. Well, the current theory in fashion is that life started as a chemosynthetic organism, that is, organism getting its energy from chemicals deep below the surface in a dark, hot vent of hydrogen and sulfur. Well, that is a theory that would work well for Europa. So there exists at least one theory for the origin of life which would work for Europa. We're not on firm grounds there, but at least it's not ridiculous to speculate that life could start separately on Europa. In terms of an ecology for life, an ecology for Europa life has to have no light, no oxygen, and no organic input. We know of 3 ecosystems on Earth that meet that requirement, shown here. So we do have a plausible ecology. And here's the details of the one on the subsurface of Africa, where the energy source comes from radioactive decay, dissociating water, all occurring below ground, miles below ground. Often people talk about deep sea vents as a model for Europa. Not a good model. These organisms require oxygen, not expected on Europa. So what could we do? You think, well, we'll go into that ocean and go scoop up some water. That's not practical. There's 10 kilometers of ice. What is practical is getting a sample of the surface. And so here is what we think are cracks on the surface. So if there's an ocean, if the ocean has life, then these surface features may contain organic material or biological material from that ocean deposited on the surface. And so the mission concept that we've been working on is what we call Ice Clipper, which is where we fly a spacecraft past Europa and it releases a small, round copper ball, which smashes onto the surface and kicks up a plume. And the impact occurs with a velocity of about 10 kilometers per second. The total energy released is about equivalent to a car bomb, a convenient modern unit of energy. So 1 car bomb of energy is released, it knocks projectiles and particles of ice into the atmosphere. The main spacecraft flies through it, scoops up those pieces and brings them back to Earth. That's our second possible way of getting material that might contain frozen remains of microorganisms. The third possible way is more recent, is from Enceladus. Enceladus is one of the small moons of Saturn. It's got a jet of water coming out of its south pole. That material is spewing into space. Presumably, it's coming out from a pressurized pocket of liquid water below the surface on Enceladus. We can fly through that plume, same spacecraft design that we talked about for Europa, scoop up those materials. So we now have 3 places in our solar system where we have a chance of bringing back the dead remains of alien microorganisms. I emphasize that it's going to be dead and it's going to be micro. These are very small and dead organisms. We now want to determine if that stuff really is biological in origin. So we've got organic material that we've gotten from Mars, Europa, or Enceladus. How can we tell that it was alive? You know, if I shoved in front of you a bunch of dark brown stuff and said this came from the subsurface of Mars, was it ever alive or is it just non-biological organic material? How would you tell? How could you tell? Well, if it's like us, if it's related to us, if it's on our tree of life, it's real easy. We just amplify its DNA using PCR. We look for specific molecules that we know that characterize all life on Earth, like ATP and so on. It's easy, but then that's not the interesting case. If it's not like us, if it's alien, then that's the hard case. We don't know how to amplify its DNA. We don't even know if it has DNA. We don't know what it's got, but that's the interesting case. So our job then is to develop a general strategy for search for alien life, given that the sample is going to be dead and microscopic, right? Well, we could go back to the literature and look at what are the standard ways to search for life. Here they are. The standard way to search for life, 1, use a tricorder. This is my favorite method, and I would be happy to use it, but unfortunately, science doesn't know how tricorders work. Even more unfortunate, science fiction doesn't know how tricorders work. If you go to Larry Krauss's great book, The Physics of Star Trek, and look up tricorder, nothing. So grim for tricorders. The other approach is what I call the Justice Potter Stewart approach to the search for alien life, which is we can't define it, but we'll know it when we see it. Well, that works if it's alive, or even better if it's alive and it's big and chasing us or we're chasing them. But remember, what we're going to find in our solar system is going to be microscopic and dead. It's not clear we're going to know it when we see it. So how would we analyze that material to determine whether it was alive? I want to make a suggestion. which is based on the rather mundane observation that life on Earth is built up on a small number of components stacked up, what I call the Lego principle. And the Lego blocks of life on Earth are the 20 L-amino acids, the 5 nucleotide bases, and a few D sugars. The giant polymers of biology are built from these units stacked over again. The proteins, the RNA and DNA, and the polysaccharides, the scaffolding molecules. And I've argued that this is likely a common property of biology as well as mass-produced children's toys throughout the universe for obvious reasons of efficiency and specificity. The Lego principle that life selects molecules and uses them only is going to be a general feature. Here is the Lego blocks of life on Earth, the 20 L-amino acids, the purines, pyrimidines, the sugars, and a few fatty acids. These are the basic building blocks of everything we see in life on Earth. The point here is that alien life could use a different set and still satisfy what I'm calling the Lego principle. You need to build with a set, maybe not the exact same set that we use. Here's the most obvious example of this selectivity. Life on Earth uses amino acids, but life on Earth only uses the left-handed amino acids in making proteins. We do not use D amino acids in proteins. You can imagine a complete biology that uses the D amino acids. It's an example, life has to choose one or the other, it can't have it both ways. That selectivity is needed for efficiency. So how does this translate into a measurement strategy? Imagine taking that sample of Martian goo and analyzing it and mapping the results on some multidimensional phase space where you plot concentration versus all types of organic molecule. And this is, in a sense, a schematic cut through that phase space. If that organic material is non-biological, that distribution will be statistically smooth. It won't be flat, but it'll be statistically smooth. Molecules will appear in there based on their chemical properties. Molecules with similar chemical properties will be at similar concentrations. The L and D amino acids will be at equal concentrations. If that material is biological, for example, life from Earth, the distribution will be anything but statistically smooth. It'll be a series of delta functions. 20 L amino acids, no D amino acids. Certain molecules in very high concentrations, way out of statistical equilibrium. It does not look like a normal distribution of molecules. What might we find on another world? A similar pattern, but hopefully different molecules. This would say life, but alien life. Not the same. If it's the same as ours, well, that's life, but it's the same old, right? So that's what we're hoping to find, and this turns it into a specific measurement strategy that we can apply to dead microorganisms. I want to end by just the observation that what is this based on? This optimistic search that we're going to find something that looks like this, where we're going to find life but with a different chemistry. It's based on a view of that, go back to that image that I constructed of a thermodynamic phase space of organic molecules. Think of it as an infinite dimensional space or semi-infinite dimensional space of all possible organic molecules. We know that at least one point on that phase space is suitable for life, and that's the point where we are at. So we are at what you could call a maximum of complexity or biological complexity in the landscape of organic chemistry. And obviously we are at the maximum of that point. Evolution would force us to be at the maximum. So from our vantage point, any direction we look, it looks like it goes downhill in the fitness landscape. And that leads many chemists and biologists to conclude that that's the only way to do life, because they see in any direction you go, things get worse. But that doesn't preclude there to be a completely different maximum somewhere else, which is also above the threshold for complexity for life. And the question is, can we find that? It may be that we'll never find it by starting here and looking because it's too far away. But we might find that nature invented it over here. And I would argue that depending on where a system starts, it might end up in a different complexity maximum. And there may be many of these. There might be a whole zoo of possible biochemistries that are suitable for life out there. We can only wait and see and hope to discover them. Let me stop there, Boris, and maybe we'll still have time for a few questions. In the back here.
So, Star Trek has shown that you could have life without carbon or oxygen and water. And you say you're looking for different forms of life, different from us. So why, maybe I don't understand stuff, but why look for water? Why look for carbon?
Well, that's a good question. I wouldn't rule out that there could be Yeah, the question is, why search for carbon and water-based life? Could there be other alternatives? There could be other alternatives, but still what we're going to search for is carbon and water-based life, because in a sense, that's what we know how to search for. So we could put together a plausible strategy that life found in places in our solar system with water, we could find life there, and that's a justifiable strategy. What we might hope to find is life that's different from ours and not the same, and we begin to start understanding in a general sense carbon-based life. And maybe from that we can develop a strategy for non-carbon-based life. But right now, we would have no defendable strategy for non-carbon-based life. The closest we could come, I think, to a defendable strategy for exotic life would be searching for life on Titan. Titan is rich in organic molecules, but the liquid that's there is not water. The liquid that's there is liquid methane. And literally last week, I was involved in a committee trying to define what would be the science objectives for the next Titan mission. And the question came up, should we make the search for life, carbon-based life in liquid methane, a goal for the next mission to Titan? And the consensus, and I agreed with this, was no. We don't have a plausible and justifiable science scenario for why it makes sense to search for life in liquid methane. We have no arguable basis that we can go to a non-advocacy review board and say, we're going to search for life and here's why.
So it could be out there, but we don't know how to find it.
Exactly. We don't know how to search for it. We don't have a defendable strategy arguing for it. So we, in a sense, we need to take baby steps. And I think the baby step is search for life that's close enough to like us that we know how to search for it and how to recognize it.
Wouldn't this approach find it?
Would this approach find it? Maybe, but maybe not. Would this approach detect life on Titan? It could, because it's based on organic chemistry. And we would certainly do this kind of measurement on Titan. So we might indirectly discover it. But we can't say that we're in a good position to argue that there's life in liquid methane on Titan and that should be the basis for a mission. We can say that there's interesting organic chemistry on Titan, let's go check it out. And if further analysis of the organics lead us to suspect something like this is occurring, then that would then change the scenario significantly.
So what are the prospects for having either past life or current life on Venus?
The prospects for past or current life on Venus. Current life, the prospects are, I think, pretty slim. People speculate about life in the clouds, but I'm not optimistic of that because on Earth, clouds are generally devoid of life. In the past, Venus may have had more Earth-like conditions, water on the surface, and maybe had life. I think if it did, we have very little chance of finding any evidence of that. It goes back to the argument for Mars. There, if there was life early in its history, we have a much better chance of actually accessing that record in terms of frozen organisms or fossils or what have you. Question in the back?
Say a spacecraft returns a sample and finds terrestrial-like life. How do you know it's not cross-contamination from the spacecraft itself?
Yeah, the question is if we find evidence of life and it turns out it's Earth-like life, how do we know it's not contamination? That's a hard question, and the way you do that is by having sterile technique. You make sure that the probe and the arms and the scoops are sterilized. It's the same problem when we do field work on Earth. We go out and we take a sample. We want to make sure that that sample is not contaminated. When we drill in the Arctic, we pull up a core. We're not interested in finding bugs that came from us in that core. But microbiologists have developed techniques for doing sterile sampling, an aseptic technique, and we would apply them in space as well. But what I'm interested in is stuff that's recognizably alien, so there we'd have no issue of whether it was contamination or not. Question here?
Based on our experience with life on Earth, how much of a time window does life need in order to get started? In other words, if the planet is only habitable for a few million years,
Right.
is it possible, on the Earth, how long did it happen between the time that life was possible and the time that life was detectable?
Right. The question was, how long does it take for the origin of life, and what does the Earth record tell us about that? The oldest evidence for life on Earth is in rocks that are 3.5 billion years old. And there's real good evidence, indirect evidence for life at 3.8 billion years old. Well, the age of the Earth-Moon system, the end of the formation of the Earth-Moon system is 3.9, 3.8. So it appears that the Earth finished its accretion and then very rapidly life began. And so you might argue that maybe the time window required for the origin of life is only tens of millions of years. But that's a very hard statistical argument to make based on one data point. You know, if you show up in California for the first time in your life and it's raining, you might conclude that it rains here every day, but in fact, that wouldn't be correct. Or if you showed up here the first day and bought a lottery ticket and won the lottery, you might think that every day you buy a ticket, you'll win the lottery. So it's hard to do statistics based on one example. And that's why it's such a powerful result to find a second example in our solar system. The difference between one example of life and two examples of life, statistically, is very, very important. It does more than just double our understanding.
Just wondering, wasn't Antarctica warm in the past? Does that give an unfair advantage to the green goo?
Sorry, say that again?
Antarctica was warm in the past, so the green goo having an unfair advantage against the Martian counterpart.
Yeah, the question is about Antarctica, which was indeed warmer in the past. The green goo we see there, though, is not isolated in Antarctica. It is in equilibrium with the rest of the biosphere. Antarctica is not isolated with respect to microorganisms. It is isolated with respect to large organisms, but the algae we find in that lake is the same algae we find out in the pond here near the bay. Algae are distributed globally, so that it is not an isolated environment in which life originated. So its application to Mars is only ecological. In terms of the origin of life, Antarctica is not telling us anything about the origin of life on Mars. We don't know how life started on Earth. We don't know where or how it started. And that really limits our ability to predict where and how it would start anywhere else.
What about the last pictures about some kind of gullies in the canyons on Mars? Like five years ago, they didn't have anything, and after 5 years, they
Right.
discovered some kind of, I don't know, some kind of liquid sign.
Yeah, the question relates to these recent images from Mars Global Surveyor, where they took pictures of certain craters, and then 5 years later, there were streaks on the side of the crater that look like water stains. And one explanation for these streaks is water, which is very surprising because we don't expect water on Mars today. There's other explanations for these streaks. They could just be the movement of dry material downhill, so-called mass flow and mass wasting. But, and then the report that was published lists all these different hypotheses. Of course, the press focused on the liquid water as the explanation. So we really have not found non-controversial evidence for liquid water today. There's lots of hints that it might be there, but it's not solid. Okay, I think maybe we should quit there since we're close to our ending time.